Inspirational personalities have achieved more than most. But we forget they are normal people, just like us. So we want to learn about the way they think, to understand how they have achieved greatness.
So today we have Madeline Lancaster, and she's a developmental biologist and a group leader at MRC Laboratory of Molecular Biology. She's known for developing cerebral organoids to study brain development and diseases. And she has significant honors, including the Eppendorf Award for Young Investigators, the EMBO Young Investigator Award, and as of recently, she's an EMBO member. She's also won the award ISSCR, Dr. Susan Lim Award for Outstanding Young Investigators. She has given a TED Talk, and she's given an interview for the Financial Times. It's a pleasure to have you here, Madeline, and thank you for coming.
So I wonder if we could just begin with you telling us a little bit about your background, where you grew up, your journey to becoming a scientist, and your career.
Sure. So I grew up in Salt Lake City, Utah, in the U.S. My mother is a psychiatrist and my father is a scientist. So now studying the brain from a sort of biological perspective really makes a lot of sense. I'm sort of an intermediate between my parents, I guess. And so I also grew up being surrounded by science and visiting my dad's lab, actually, and actually saw a neuron the first time in his lab and was sort of, just amazed by it. And I mean, anybody who's actually seen a neuron through a microscope, I think, can appreciate the beauty of it and the complexity. And so I just sort of became intrigued with how these beautifully complex cells lead to all of the amazing abilities of our brains. And that was then something that I sort of took with me all through my education. So even in high school, I was really fascinated by psychology, also probably because of my mom's influence, and also spending time with her in her clinic, actually, and seeing some of her patients in the waiting room and stuff. But then with psychology, I was a little bit left sort of, I was just left wanting because you understand it on a very superficial level, but I always wanted to really gain some sort of mechanistic understanding of really, why are these people, for example, with schizophrenia experiencing, hallucinations, what's really actually going on at the cellular level within the cells. And so then I actually then in my undergraduate work, where I got a bachelor's at Occidental College in LA. So I left Salt Lake City, moved to LA, got a bachelor's in biochemistry. Because, as I said, I became interested in that sort of fundamental mechanistic insight. And then after that, I decided to get back into the brain, though, and I joined, during my graduate work, I joined the lab of Joe Gleason in San Diego, where I worked on a neurodevelopmental disorder called Joubert syndrome. And I was working on this using a mouse model, and that then actually got me really into developmental biology. So I've always had this interest in the brain, but not necessarily development. And in fact, I think in college I hated development, to be honest. But once I got, started really studying it and researching it, I really fell in love with understanding how tissues build themselves and how cells know where to go and what to become. And so that's when I then, for my postdoc, decided to go to Jürgen Knoblich's lab, who's in Vienna. So that was quite a big jump, moving across the ocean. But he had and has a beautiful way of looking at developmental biology, really trying to gain basic mechanistic insights. And traditionally, he had been working in flies. I really appreciated the fundamental insight that his lab was gaining in flies, and so I thought maybe I could bring my experience in mouse models and apply some of those basic principles to the mammalian context. So then, when I started in Jürgen's lab, we had this idea together that we would take some fly genes and we would start looking at them in the mouse context. There was a little bit of an interesting story here where because it took me a while to actually move. It was such a big move, so I kept pushing off my start date. And by then, several new postdocs joined in Jürgen's lab, and they actually took all the best genes. So I got to Jürgen's lab, and there were a whole bunch of sort of like leftovers, like hundreds of genes that had come from a fly screen, but it wasn't, the top candidates had been taken. So we're left with this list of around 600 genes. And I sort of said, well, I'm not going to go and start making 600 mouse mutants. So let's try to find a way of screening through these. And so I proposed to Jürgen that we start using this very strange new in vitro model called neural rosettes. They were brand new at the time. And I'd heard about them in San Diego from Rusty Gage's group there. And so that's kind of actually how I got started on what really set my career in motion. So working with these stem cell derived neural rosettes, actually then that turned into what we now call brain organoids. And so then, yeah, in Jürgen's lab, I developed these brain organoids that I then used to study, which we then used to study a disorder called microcephaly, but we're now using in my lab to study really fundamental aspects of brain evolution. So sort of getting back to that thing that got me excited in the first place, which is like, what makes our brains so unique and special and gives us this amazing cognitive ability, but also makes us highly susceptible to, these devastating disorders like schizophrenia. And so I think an evolutionary perspective there, an evo-devo perspective really
Evo-devo. I've never heard that.
is what I think is what my lab is now doing. Yeah.
Could you tell us a bit about the process of discovering that these cells could clump together and form these interesting organizations? Because I think it's a fairly unusual story.
So it's a classic sort of story of serendipity. Basically, I mean, if I had joined Jürgen's lab a few months earlier, then I would have taken one of those great genes and I wouldn't have done what I ended up doing. And so it was very lucky from that perspective. It was also lucky that we decided to try to move in vitro because we had all of these genes and there was no way to do like a screen in vivo, at least not at that time. And also, I had 2 other projects at the same time. So I had actually 3 projects when I started in Jürgen's lab. I was very ambitious, I guess. One was, I did have a list of about 3 genes that we were particularly excited about, but they didn't really work. So I guess that was also serendipitous that didn't work.
So a lot of failures, really.
And then, exactly. And then I had another project that I was really excited about because in my PhD, I had worked on cilia because Joubert syndrome is a ciliopathy. And I wanted to look at the role of cilia in early neural development. And that actually, I had some nice data, but everything else took over. So there's this data that we never actually even published. Because basically then with this in vitro approach, I decided to take a really sort of simple approach initially, probably because I'm not a stem cell biologist really. I was working with mouse embryos. I decided I'll just take some mouse embryos, I'll dissect the forebrain, the early neuroepithelial cells of the forebrain, and I'll just plate them down and try to make neural rosettes that way. And in Jürgen's lab, they didn't really do much in the way of tissue culture, really at all. So I found some old laminin and fibronectin. These are used to coat the dish so the cells will stick. So I used that to coat the dish, and it just didn't, I think it was old. So what ended up happening is the cells didn't really stick. They instead just remained, floating in the media and tended to stick to each other. So then I ended up with these really cool blobs of cells. Exactly. And I just remember looking at them and thinking, wow, that really looks like a neural tube, like the precursor of the brain. It's like a round thing with a space in the inside. There's organization. The cells are like lined up nicely. I thought that was really amazing. So yeah, from there, I kind of went down a little bit of a rabbit hole. I remember giving lab meeting, and at some point, I think it was Jürgen actually was like, what are you doing? I thought you were supposed to do a screen.
But I've got these blobs.
Yes, and I was showing these blobs, like, these are so cool though, don't you think? And I think, but I think I managed to convince everybody that it was worth doing this when I started showing some of the pictures of where you could really see neural progenitors and then the neurons being produced in a very specific sort of spatial and temporal organization, just like you see, in the actual embryo. And then, of course, I think everybody realized how exciting this was, and we took it from there.
So did they kind of see it as quite a big risk, or were they all as excited as you are? Did it feel like a very cooperative in that way?
So in the beginning, I was definitely sort of like a lone ranger lab or something. Sort of, there was sort of most of the lab was still doing fly work. There were a few mouse people. We actually had a mouse club of like 4 or 5 of us. And I was in the mouse club, and we would have meetings, and I would present my work. And I think people were kind of like, well, that's kind of weird, but I mean, where's the actual biology, I think, which I understand because it was kind of developing a method at that point. But it was, I remember really vividly when I first showed the picture, and also when I even saw it for myself, the picture of the eye tissue that was forming, where you could actually see the circular structure of the retinal pigmented epithelium and the layers that actually look like an eye sort of growing off of one of these organoids. And that was, I remember hearing people sort of say, oh my God, like,
They got it, so it was like
there was a switch, yeah. Yeah, so, and then it was, I think people realized
We're on board.
how much potential these things actually could have.
Because to me, this is what science should be, right? It's like no expectations, just seeing something interesting and just following where it takes you. But I think a lot of young scientists feel like there might not be space to do that in a lot of science. So I wonder if you could talk about if you think that that's a problem that we have in the field of science at the moment, is that there isn't the space for you to just meander down these paths and see.
If my cells had stuck the first time, and I had made rosettes the first time, because eventually I did. I even did a little mini screen of 96 siRNAs that I was knocking down, and it worked and everything. But of course, again, another example of something that we sort of put on the back burner because everything was working so nicely with the organoids. If that had worked, I probably wouldn't have done this. And also, even if let's say it hadn't worked, but it hadn't worked after I'd already been there for 2 years, I wouldn't have really had the freedom to explore. It was good that it happened. I mean, I joined Jürgen's lab in early November 2010. The first organoids I made were in December.
wow.
Like the first mouse that I dissected and plated and failed to stick and formed these aggregates, that was in December. So it was so early in my postdoc that I didn't have the stress of trying to just get a paper published and get out, right? So I think there's so much about luck there.
Yeah.
And I think it's a shame that you have to rely on luck for that. So having freedom to just kind of play, I think is really, really important. And I guess part of the reason I was also, because then of course it took me like a year and a half just to work out the culture conditions and get it working reliably and really get it working from human cells and all of that.
So there's a lot of trust that they had.
There's a lot of trust they had to put in me. And this was, none of this was like through, none of it was planned, right? So that means there wasn't like a grant that was specifically written to do this, because that would have been crazy.
Yeah.
To propose, I'm going to make human brain tissue in a dish. Like nobody would have funded that. That's crazy. You can't make human brain tissue in a dish. But so because IMBA, where I was in Vienna, is a core-funded institute, that gives you freedom to explore much more. So I think that's also an important aspect.
Well, that's exactly what I was going to ask you. Is that a big part of the reason why you were attracted to the LMB specifically, which has its own reputation in this regard?
Yeah, so exactly. So then when it came time for me to apply for my own position, I mean, IMBA and its sister institute, the IMP, they're sort of together. They both of them are very much influenced by actually the LMB. The LMB has served as an influence for a lot of different places, including like the EMBL in Heidelberg, Genelia in the US, lots of these kind of core funded places. And the reason is because it is such a successful model, because you give people who have proven themselves to have good ideas, you give them the money they need, and you let them
be free.
play.
Yeah. Yeah.
I think that's really, really important. So yeah, when I was looking for my own position, I had offers from lots of different places. I could have gone back to the States as well. There aren't very many places in the States actually that work like that as well. And also, yeah, I got spoiled at IMBA, like the media kitchen, like not having to make PBS or I don't know, anything really. And not having to worry about like in my PhD, having to worry about how many hours I was costing my lab every time I went on a microscope, At the LMB, I don't have to worry about that. At IMBA, I didn't have to worry about that.
And it is an element of sort of spreading yourself a little bit thin, because you did say you had a lot of projects on, right? So
Yeah.
the more you have, that means you kind of like switch a mindset from one to another, and you're more likely to kind of want to explore different things if there's no like specific pressure
Yeah.
to just publish like as soon as possible.
Yeah.
So I guess for you, it was early on.
Yeah, I think it's good. I mean, this is something that I do with people in my lab as well. When they start out, I give them a couple of things to get them started, so that they can, because it's hard to predict what will work and where things will go. And I'm happy to kind of go where the science takes us. So if we have a particular hypothesis, and it turns out to be wrong, that's great. There's something interesting there. Then let's follow it where it leads.
Yeah, so in some of your previous interviews, you've talked about how you saw the clumping of cells being something specific. But, I think most scientists wouldn't necessarily follow that up.
Yeah.
But you had the courage to just do it.
Yeah, so I think, yeah. So I think one thing so in the beginning, when I would talk about this story, I chalked it all up to serendipity. I think I've learned in recent years that I should also take a little bit of credit there. And I think often actually women are particularly less likely to take the credit they deserve. So I feel like I should take some credit there. And I think what I can take credit for is that that I did recognize that there was something interesting going on there. And in particular, I went to a stem cell conference a few years after. And again, I'm not a stem cell biologist really, although I'm sure everybody would say that I am now. But at least at the time, I was certainly not one of those core sort of stem cell people who was trying to, let's say, make dopaminergic neurons from human pluripotent stem cells. So I went to one of these conferences and was speaking with somebody who is one of those core figures in the stem cell field, and was telling him this story about how I had these cells that had clumped unexpectedly, and that's what ended up leading to organoids. And he actually said, "Oh yeah, I've seen that before." I was like, "Well, what did you do then?" And he said, "I threw them away. That's not what I was interested in. I was interested in making pure dopaminergic neurons. I didn't want anything funny that wasn't, the right identity and doing what I wanted it to do." So I think also probably, and this probably comes from me not being one of those people that was like really embedded in the field, and rather coming from that developmental biology perspective, just kind of being really interested in nature, in biology, in what cells can do, and letting them do whatever they want to do, and then using that to teach us something.
I guess there's also an element about inexperience that kind of makes you want to
I was going to say exactly the same thing that inexperience can convert to.
Exactly. I think you see that time and time again. There's you got to have the right balance, but a little bit of naivete and and curiosity and creativity too, I think is really important.
So I guess aside from curiosity and creativity, which they do tend to go together for scientists, is there something distinctive about your personality that you think helped you throughout your career or personal life?
I guess maybe kind of going against the grain a little bit. I think it's also really important though to point out that I have the privilege that I've had of having grown up with a scientist father, and a mother who she also has a PhD, actually, although then she ended up getting the MD and becoming a psychiatrist. So that's really, really important to recognize, that being surrounded by scientists and people who think like scientists, some of it may be innate and genetic, but a lot of it is definitely also cultural and environmental, and that does definitely give me a privilege. So I'm not afraid of scientists because my dad is one, and when your parent is
my particular, like, it makes you feel like it's possible for you to do. It's not a,
and they're not scary.
Yeah.
Like, they're just people, because my dad is my dad. Like, so I don't, whatever famous Nobel Prize winner, I mean, they're just a person too. So
It also sounds like you don't mind challenging things. So that's not so showing that, but so I want to challenge you on this, like actually tell me about it.
That is definitely, and I think if you talk to my PhD supervisor, he would definitely agree that I challenged him quite a bit. Yeah.
Would you say that the fact that you grew in such an environment sort of influenced your idea of what success will look like for you personally?
Yeah, I mean, I think, I remember, let's see, when I was a postdoc, for example, and talking with colleagues of mine, like, what do you want to do after your postdoc? I want to be a PI. I want to have my own lab, learn math. How long have you known you want to do that? Since I was a kid, because that's just, I've never questioned it at all. And so I think just having that always sort of throughout my life, knowing that was my goal, that's where I wanted to be. And I was happy with, I always thought, well, okay, I can't control where I publish. It's really hard. I can try my best to publish an important paper, but if the reviewers hate it or something and it ends up not being in a top tier journal, at least I can try to do some, to contribute some valuable science. And then, I'm sure that there are small universities somewhere out there who are looking for PIs, even with, with a paper like that. And I'll take that if I have to, because I just really wanted to run a lab wherever. So yeah, I didn't necessarily know, like, you can't plan to, like, make a really big groundbreaking discovery necessarily, but I think I always figured, well, I'll just try my hardest and try to contribute something that's where I feel like it's worthwhile. Ideally, where it contributes to changing our fundamental understanding of biology. And if it's really, really important, it will be recognized as such, I think.
It's interesting because for you, it doesn't seem like it's about prestige, right? It's about having just the environment where I can actually pursue what I want to pursue. So if somebody gives that to me, I'll take it.
I think for me, if you'd ask me, like, what my goal is, like, for all of this, I think, for some people, it's like, I want to cure disease, or I want to understand human disease. And there's always been a little bit of that, but really deep down, fundamentally, I want to learn something completely new that, like, changes textbooks. Like, where it's like, okay, the textbook says this, but now because of work I've done, we now can say this, that it's like we've truly contributed to our knowledge of how the brain is built and functions. And, whether it gets published in Nature or not, like, ultimately, I think what's important is that it's an important contribution to our usually, if it's a really big, important finding, it will get published, well as well. But yeah, the main goal for me is that it really changes our understanding of biology.
So what about a personal trait that hasn't served you so well in achieving the goals that you want to achieve?
Let's see. Well, I'm kind of messy. I'm a little bit scatterbrained. And I still have a bench in my lab, which my lab members may not be happy about because it's probably the messiest bench in the entire lab. There's always, like, tips and things scattered around, and so I often have to. It's probably not very efficient in terms of time. The amount of time that I end up wasting because I have to reorganize something because I haven't done it, properly in the first place. So I think that's something that, yeah.
Well, that's very encouraging for me personally. So exactly like that. And what about something that you enjoy doing outside of the lab that people might not expect?
I enjoy a lot of things, actually, when I have time. So I have 2 young children, so I don't have as much time for my hobbies as I used to, but they're starting to come back. My kids are getting older and starting to have some time again. So I like to paint, actually. So I like to paint and draw. I do like pen and ink drawings. But probably what maybe most people might not expect is I love playing video games. So my favorite right now is the new Zelda game. I love it. So any chance I get, after the kids are in bed, I'm playing Zelda.
Do your kids play video games?
A little bit, yeah, but
So you get on their
still
console.
yes, so I'm playing on their Nintendo Switch. Yeah, so I play Mario Kart with them, and then after they go to bed, I play Zelda.
Yeah, that
So to it, they're not quite old enough to really like Zelda yet.
Just to sort of switch gears a little bit back towards the science, we're just curious, since you're a pioneer in the field, where do you see this field going? What people could do
Yeah,
with your discoveries?
yeah. So, I'm using it to try to gain fundamental new insight, but to be honest, I'm a minority really in the field in terms of the way I'm using them. Most people are using organoids, in particular brain organoids, to model human disease and to even start like testing drugs or other types of therapies. And so I think we're going to see more and more of that. I've already heard of a few companies that are starting to develop therapies based on organoids that are starting to enter clinical trials, which is really, really exciting. So to know that a model that I developed 10 years ago could actually lead to a therapy for something, for a condition, a human condition, is particularly rewarding, I have to say. It's funny because it's one of those things that was never my goal, but if it actually happens, like the thought of it, it really, really does make me feel very good about what I've contributed. I think we'll see more and more complex organoids, different types of organoids, regions being put together in these so-called assembloids, where you're sort of fusing different parts of the brain, or even bringing in other like extra CNS tissues and seeing how they interact, like for example the gut-brain axis, like looking at how
Oh, I never thought about that.
signals from the microbiome could influence brain development and function. So I think we'll see a lot more complexity being built in. At the same time, I think we'll also see even simpler organoids too. So I think we kind of started with something that's sort of in the middle, and we'll see things branching in all different directions. Because also simpler has an important place too, especially for drug testing and stuff, where you really want to know like exactly what cells are affected by the drug.
You seem to have a very positive view about the future of this. Is there anything that makes you nervous about these discoveries and where they could potentially go?
I'm not too nervous really at all about the actual organoids or their use. I think it's pretty clear that they have only positive things to contribute, in my opinion. I'm a little bit concerned about sometimes the way they're portrayed by some people, some scientists who are using them, but particularly I think sometimes the media, the way that it can sometimes be portrayed in the media, or in other sort of like popular press, where I think or also even by sometimes by very well-meaning ethicists, but they sort of blow out of proportion sometimes what organoids really are, and kind of start sounding alarm bells when they shouldn't necessarily be sounded. So I think it's important to be very clear about what brain organoids actually are, that they are, as I said, they're not actual brains. They don't have the kind of fully formed structure and interconnectivity that you would need to have a functioning brain. They're tiny, and they don't have a body. So, I mean, a brain without a body is really nothing, really. So I think there's lots of things that sometimes people try to, say about organoids, that they're concerned that they could be conscious or something. But, I think it's really important to keep in mind like what they really are and what they really aren't. And what concerns me about the reason it concerns me that sometimes this can be portrayed inaccurately is that can scare people, which I understand. If I didn't know anything about brain organoids and I read some of the things that have been published about organoids, I would be concerned because I'd think, oh my gosh, these are like brains in a vat.
Yep.
And it's like those like
Floating
sci-fi
brain,
movies or something, where it's like a floating brain. The poor person who is that brain, they're like, let me out, that's awful, right? So if people, of course, think that that's what we're doing as scientists, then rightly so, they're going to be concerned and they're going to want it to stop. But if they want it to stop, that will stop all of the positive directions that these organoids can contribute to. So what I like to remind people of is that there are millions, hundreds of millions of actually conscious human beings out there who are suffering and need treatments, new treatments. And we have no new treatments for schizophrenia. Like schizophrenia is being treated with antipsychotics that are like 40, 50 years old. Nothing new has come on the market because the animal models are not sufficient. The existing in vitro models are not sufficient. So if these organoids could lead to new treatments for those patients, that would help actually truly conscious beings who are suffering. And this, I think this hyping of organoids and making people worried they could be conscious, I think is not, it's not good for anybody really. So I think, yeah, we need to be careful about how we talk about them.
So we know that you have an interest in science fiction literature, or at least we've read that. Correct us if we're wrong.
Yeah.
So we wondered, did this interest precede working in an area that the public at least perceive as kind of as close to science fiction as we can get? Or do you think it kind of developed from you working at the frontier of technology?
That's a good question. I actually never really thought of there being a connection there in my own with the research, but it is actually, it's very cool that, that sort of what I'm doing was science fiction and is now becoming science fact. And I mean, I think, I still love reading science fiction. I love to, I'm always looking for new science fiction to read, and it's usually more on the side of sort of like space travel type science fiction. But I think some of the things I've read recently, like from Margaret Atwood or from Kurt Vonnegut, like sometimes it gets into these kinds of very interesting, also ethically sort of questionable areas. So it's cool that I can bring to life a little bit of science fiction. Yeah.
Is that like an interesting sort of mental exercise for you as well? Because you can kind of think just in a scientific way, science fiction way, where could your research go? And it's probably fun in that way.
I think it's fun to sort of I think it helps stimulate that creativity that I think is so important. So maybe not necessarily, like I said, we never set out to build brain tissue because that would be crazy. But I guess sort of looking at it and saying, well, this is very interesting. Maybe there's something, like, especially looking for something weird, right? Like I'm always on the lookout for something weird happening, because that's usually where something interesting, something new, where you really gain new insight. So maybe that also comes from liking science fiction and liking the weird sort of side of that type of literature. So
yeah, on the topic of the insights that you've got from the organoids, we wanted to ask, on a sort of personal level, have your organoids changed at all the way you think of the brain or the way you think of human cognition, or has it all been sort of fairly what you would have expected?
Yeah, it's definitely changed the way that I think about human cognition. So one thing that's really changed my thinking is our discovery that that what really seems to set apart the human brain from other apes is that it takes longer. So we've found that everything is happening much slower in the human developing brain, and that's why it has a chance to grow much bigger. And I have 2 young children, and so when I sort of think about how they're developing in terms of their, like, reading or writing or maths or whatever, and so it sort of makes me think about when I'm thinking about how they're doing benchmarking compared to like where they should be with their peers and that kind of thing. I think it makes me think, well, maybe actually being a little bit slower might actually be a good thing. and that the end point might be further, might actually be greater than otherwise. So it's this kind of emphasis that we as a society also place on like precocious children who like already play Bach when they're 4 years old. Is that, I don't know. I mean, maybe also the slower children are actually, they end up at in a place where they have even higher cognitive potential or creativity or whatever. So I think it just makes me realize how we need to value that sort of diversity. And yeah, if I was going to make a super smart human, I would probably make one that went even slower, because then it would have an even larger brain and even more complexity in terms of neuronal, network formation and stuff.
That's reassuring to hear, considering how fast-paced the world is becoming. You actually think that on a cognitive level, even slower is better. So that's nice to hear. I guess to sort of end our discussion, we were curious, if you weren't going to be a scientist, what else would you have been?
Oh, that's a good question. So I remember when I was a postdoc or a PhD student, I remember some days looking out the window and thinking, oh, today's really stressful. That looks really fun. And looking over at the construction workers actually cross the streets because they're outside all day. They go home at the end of the day and they relax. And I thought, that would be nice.
Well, you kind of are a construction worker, right? You're just building something else.
Exactly, I'm building something else, and I love building stuff. I play Legos with my son still. I build all the furniture in the house. I do all those sort of. So I like to use my hands. So I don't know, some kind of building or something like that. But I'd like to be outside a little more. That's the only thing I kind of don't like about this job, is that I'm inside.
So that is really interesting. There are so many threads of this that kind of help it make sense why you were the one who maybe saw this and thought that it was worth taking up. The fact that you're a little bit more of a renegade and you don't necessarily feel the pressure to follow the herd, and you like building things and constructing things and playing. So final question that we'd like to ask is, what's the one lesson that you think that you've learned in your life that you would like to share?
Yeah, I think it's just really important to try to take the pressure off a little bit. I think that's the lesson I would like to share with people, young scientists who are maybe starting out or sort of midway through, to try to remember to take the pressure off and try to remember that this is supposed to be fun. Really, science is really fun. And so if it's not fun, then something needs to change. So yeah, I would say just find whatever gets you most happy, where you find the fun, and do that. And I'm sure that you will make an important discovery.
Thank you so much for joining us, Madeline. It's been a really nice discussion.
It's lovely meeting you.
Thank you.